When we talk about diseases like Alzheimer's or Huntington's, one of the biggest roadblocks we face is diagnosis. We just don't have those clear biological signposts or biomarkers to track them effectively. But what if machine learning could change that? What if we could build a model to scan the very proteins at the heart of these diseases and tell scientists, hey, look right here? Well, today we're diving into a new predictive model that aims to do exactly that, and it's a potential game changer for how we approach these devastating conditions.
All right, so here's how we're going to break this down. First, we'll get a handle on the core problems scientists are up against with biomarkers. Then, we're going to explore the three major breakthroughs in this model. First, a much sharper way of seeing proteins in 3D. Second, a really clever method for training a model on messy, real-world data. And third, an intelligent search strategy to find the signal in the noise. And finally, we'll see how it all comes together and what it could mean for the future.
You know, neurodegenerative diseases affect millions and cost our healthcare systems billions. And a huge piece of that puzzle is the lack of reliable biomarkers. These are the measurable signs that can tell us if a disease is present or how it's progressing. Without them, finding answers often means relying on incredibly long, expensive, and sometimes inconclusive lab experiments.
Now, at the very heart of this problem is a biological process called phosphorylation. The easiest way to think about it is like a tiny light switch. When a phosphate group gets added to an amino acid, it flips a switch on a protein, changing how it behaves. Now, for a healthy brain, a protein called tau needs these switches to work just right. But in diseases like Alzheimer's, these switches can get stuck in the on position. This causes the tau proteins to misbehave, clump together, and become toxic. So, being able to predict where these switches are is absolutely critical.
And this number right here, 135, that really shows you the scale of the problem. On that one key tau protein, there are 135 spots where these switches could be. But here's the thing, researchers have only ever managed to confirm 43 of them in the lab. Finding the rest is a classic needle-in-a-haystack situation, but on a molecular level.
And that, right there, is why computational models are no longer just a nice-to-have. They are essential. The source material says it all. Trying to find every one of these sites in a lab is, well, a colossal challenge, if not downright impossible. We need these smart models to narrow the search and point scientists toward the most likely targets. Okay, so if we can't just rely on lab work, how does a computer even start to help? Well, the first big step and the first major innovation here is all about creating a better map. How do you actually represent a protein in a way that's accurate enough for a computer to predict its chemistry? And here is that first huge leap forward. The old way of doing things was kind of like using a blurry map. It located an amino acid using just a single point, its central alpha carbon atom. But the new method is so much smarter. It calculates the center of mass using all the atoms in the amino acid's side chain. That's the part of the molecule that actually does the reacting. So you go from a rough guess to a high-resolution blueprint of its exact chemical position. It's a game changer for precision.
But knowing the exact location isn't quite enough. A molecule's behavior is all about its neighborhood. So the model doesn't just look at the target amino acid by itself. It draws 6 concentric spheres around it, like ripples in a pond. Then, it identifies every other molecule in each of those spheres and gathers up all their chemical properties. From all that data, it generates 192 distinct environmental features. This gives the model a super rich 3D picture of the entire chemical environment.
And what kind of properties are we talking about? Well, it's all the stuff that really dictates how molecules behave. Things like polarity, which is its tendency to hang out with water, its ability to form hydrogen bonds, its size and shape, its surface area, and even how flexible it is. This whole chemical profile gives the model a deep, layered understanding of the forces at play in that local environment.
Okay, so we've got a really precise 3D map. But any machine learning model is only as good as the data you feed it. And that brings us to the second major innovation. Because in biology, your data is pretty much always messy and unbalanced.
Right, this is the next massive hurdle. The data you have to train on has 2 big problems. First, it's incredibly imbalanced. You have way more examples of sites that are confirmed negative for phosphorylation than positive ones. But the second problem is more subtle. A negative label doesn't always mean no. It often just means we haven't proven it's a yes yet. So you're dealing with this huge ocean of uncertain data. If you just train a model on that, it's going to be biased and not very reliable.
So how do you clean that data up? This is where they got really clever. They developed a confidence score to sniff out those unreliable negatives. Instead of treating every negative example the same, they put each one to the test with two key questions. First, do other prediction models think this site might actually be positive? And second, using a measure called Mahalanobis distance, how chemically similar does it look to the sites we know are positive? If a negative sample gets a high score on this quiz, it's flagged as unreliable and kicked out of the training set.
And this Mahalanobis distance is a really cool tool. Let me break it down. A simple distance might just compare two things on one variable at a time. Mahalanobis distance is way smarter. It understands that variables are often related. For example, it knows height and weight are correlated. By taking into account the relationships between all 223 of our chemical features, it gets a much better feel for the true shape of the positive data. It makes it much better at spotting those sneaky, unreliable negatives that are trying to pass themselves off as the real deal. So, after that filtering step, we've got a much cleaner set of negative examples. But there's one last move. To make sure the model learns from a really diverse set of examples, all those reliable negatives are grouped into 8 distinct clusters. Think of them as 8 different types of negative sites. Then the model is trained on the most representative sample from the center of each of those clusters. The result is a training dataset that's not just clean, but also incredibly diverse, teaching the model to recognize all kinds of negative sites. So we have a precise 3D model, and we have clean, diverse data. This brings us to the third and final innovation, which is all about tackling complexity. Remember those 192 environmental features, plus others? In total, we have 223 features for every single data point. How on earth do you figure out which combination of those is the most powerful without getting totally lost?
To put this in perspective, the number of possible combinations of those 223 features is 2 to the power of 223. I mean, that's a number so huge it doesn't even have a name. It's functionally infinite. Trying to test every single combination to find the best one isn't just a bad idea, it's computationally impossible. You need a computer the size of the solar system, and you still wouldn't be close. You need a much smarter way to search.
So the search begins by creating a kind of hit list. Each of the 223 features is given a pertinence score. This score comes from running 4 different statistical tests, and they all ask the same basic question: how good is this one feature, by itself, at telling the difference between a positive and a negative sample? This creates a ranked list, like a heat map, showing the algorithm which features are most likely to be important from the get-go.
So, to search that infinite space, the model uses a really cool strategy inspired by how a blacksmith works metal, called simulated annealing. It starts off at a high temperature. It's in full-on exploration mode, trying all sorts of random combinations of features, even ones that don't seem that promising. Then, as the system cools down, it starts to get more focused. It balances exploration with exploitation, starting to favor the features with those high pertinence scores. Finally, at a low temperature, it goes all in, focusing almost exclusively on the highest scoring features to fine-tune the absolute best combination it can find.
So, after all of that, a more precise 3D model, a smarter way to sample data, and this intelligent search for features. What was the result? After all this meticulous work, how well did the final model actually perform?
Well, the results really do speak for themselves. When you put it up against other top-tier models using the same standard tests, this new model comes out clearly on top. The metric we're looking at here is the Precision Recall Area Under the Curve, or PRAUC, and this model hits a solid 0.90, which is a significant jump over the competition. And here's that final score, 0.9015. In the world of machine learning, a score like this is a really big deal. What it means is that the model is excellent at finding the true positive sites, the ones scientists really care about, without raising a ton of false alarms. In other words, it's both accurate and reliable.
But this was never just about getting a high score on a chart.
This is about the real-world impact. When the researchers turned this model loose on the protein associated with Huntington's disease, it pointed them to 5 new high-potential target sites that had never been experimentally confirmed. This is the ultimate goal: to create a data-driven roadmap that helps scientists in the lab work faster and smarter.
This whole project is just a fantastic example of how creative and rigorous machine learning can help us untangle some of biology's most complex knots. And it really leaves you with a pretty exciting question. As these AI tools get even better at decoding the language of life, what other incurable diseases are they going to help us unravel next?